has to use one word. Good evening, folks. I'm Dr. Jeff Pribble from the Department of Biochemistry, Chemistry, and Geology, and I'm your host for today's event. This is our 23rd Leonard A. Ford Lectureship. Dr. Leonard Ford was a professor of chemistry at Mankato State College from 1939 until his death in 1967. He was chairman of the chemistry department from 1939 to 1947, chairman of the Division of Science and Mathematics from 1947 to 1965, and coordinator of special programs and chemistry professor from 1965 until his death in 1967. Dr. Ford made significant contributions to science education during his tenure at Minnesota State Mankato. He published articles in journals such as Science Education, School Science and Mathematics, and the Journal of Chemical Education. He also published articles with the American Chemical Society and the Minnesota Academy of Sciences. His book, Chemical Magic, was first published in 1959 in hardback and later in 2 editions of paperback. This book also achieved international readership, being published in French, Spanish, Danish, and Japanese. Leonard Ford established the science fairs at Minnesota State in 1951, and the science fair continues to this day as one of the largest science fairs in the state of Minnesota. Dr. Ford was instrumental in establishing the nursing program, medical technology program, dental assistant program, dental hygiene program, and engineering programs at Minnesota State Mankato. Dr. Ford was recognized by his professional community as a leader. He was president of the Minnesota Academy of Sciences. He served on the education committee of the American Chemical Society and was committee chairman, science section of the Minnesota Education Association. He was president of the Mankato Lions Club and served on the board of trustees and alumni board at Gustavus Adolphus College. This lectureship receives financial support from the Ford family. With us this evening is one of the daughters of Leonard Ford, Dr. Barbara Ford Olson, and her daughter, Kathy G. Please join me in thanking them for their support. Dr. Ford's legacy is truly worthy of celebration. This year, I'm pleased to introduce Dr. Christian Abnet. Dr. Abnet is Chief of Metabolic Epidemiology Branch and Senior Investigator at the National Cancer Institute, which is part of the National Institutes of Health. Dr. Abnet earned his Ph.D. in environmental toxicology from the University of Wisconsin-Madison and a master's in public health in epidemiology from the University of Minnesota. He has spent his entire career at the National Cancer Institute. In 2014, he was awarded scientific tenure by the National Institutes of Health and named chief of the newly established Metabolic Epidemiology Branch. He serves as the principal investigator for multiple large epidemiological studies in the US, Asia, Africa, and South America. He is also on several committees of the World Health Organization. He is an elected member of the American Epidemiological Society. Dr. Christian Abnet, welcome to Minnesota State Mankato.
Thank you, Jeff. Good evening. Can you guys hear me okay? No? Try this. Hopefully that's a little better. Okay, great. So I'm very happy to be here today, and I'm a little bit nervous for one particular reason, and that's because my mother-in-law and my father-in-law are in the audience. My father-in-law was a professor of chemistry here at Mankato State, and I don't think I've ever spoken in front of them before, so please give me a little leeway. So I'm going to talk about my work in cancer epidemiology and how we describe the burden of cancer, investigate the causes, and then try to discover the means of preventing cancer. Normally, at biomedical research talks, we give disclosures so that we understand where everybody's coming from financially. I'm a federal employee. I cannot accept money from anything other than my salary, so I don't have any speaking fees to disclose. And tonight I'm speaking as an independent scientist from the Cancer Institute. I'm not speaking for the Cancer Institute. So this is my maternal grandfather, William A. O'Brien. He was the head of the Minnesota Cancer Society and a professor of medicine at the University of Minnesota. And unfortunately, he died in 1947, long before I was born. So he never inspired me directly, but he did make me think about the importance of human health. And I started out in biology, but I ended up studying human health, I think partly because of his legacy. You can see he's sitting in front of a WCCO microphone because he was a radio doctor in the 1930s and 40s. And my uncle, William A. O'Brien II, was also a radio doctor. So when I was a kid, I used to get to hear my uncle answer questions on WCCO on the weekends. My grandfather continued to teach, even though he was an administrator, and he taught personal hygiene. So at the time, every kid starting college when they were a freshman had to take a class in personal hygiene and learn about how to take care of their own bodies. And it does sound kind of funny, and they made a movie in the 1950s using his, in the 40s, using his work, and it was called Body Care and Grooming, and it's such a silly movie that it actually ended up on Mystery Science Theater, a TV show where a guy and two robots make fun of silly movies. So I had that legacy to live up to. Okay, so as I mentioned, I started out in biology at the University of Oregon. When I went to Wisconsin, I was studying environmental toxicology, and I was studying this chemical. This is 2,3,7,8 tetrachlorobenzoperidioxin, which is one of the most potent toxins in the world. And I was studying how it affects fish. I wasn't doing anything to do with human health. I was interested in how fish were affected by an environmental toxin. I was doing molecular biology, and when I finished that, I realized I had sort of made a mistake, and I had entered the wrong field, but I already had a Ph.D. So I had to figure out what to do, and so I went into a fellowship that sent me back for one more year of school, and I went to the University of Minnesota, and I learned about epidemiology, and I realized that was the right field for me, and so I've stuck with it ever since then. I'm the chief of the Metabolic Epidemiology branch. This is a group of 12 principal investigators, and we have about 40 other staff members. We study the relationship between diet, energy balance, hormones, tobacco, and cancer. And so we're trying to understand how these factors can either make you more likely or less likely to get cancer. Training is a major part of our work at the National Institutes of Health. We have a huge group of fellows. I typically have about 20 fellows in my group. And this is a picture of the 41 fellows that have passed through my branch in the last 4 years. So you can see that it's quite a diverse group of people, except in one way, it's almost all women. This is a science that has become almost completely dominated by women. It's not really clear why. It's mostly a biostatistically oriented work, and for reasons that I can't explain, we draw a lot of women as trainees. So far, from this group, I've had 14 people in the last 4 years who went on to become assistant professors at universities. So I think we've been really aimed at training the next generation of public health scientists. So, thinking about cancer research, you can think that there's two kinds of cancer research that a lot of people think about when you hear that word. And the first one is called clinical research. This is physicians working with patients trying to figure out how to treat their cancer and either cure it or at least keep them alive. And here's an image of a little kid. Unfortunately, he's got a childhood cancer, and he's hooked up to some machines, and they're trying to work out new ways of treating cancer. And that's the most important and the most well-funded part of cancer research, is directly trying to do therapy. The other aspect of cancer research that a lot of people are familiar with is basic science bench scientists. So here we have an image of a young scientist standing at a bench. She's got pipettes and test tubes and flasks, and she's probably working in cell culture to try to understand the basic biology of how cancers form, how tumors evade the immune system, and so forth. But there's a third aspect, and that's what I do, and that's what we call epidemiology. This is the branch of medicine which deals with the incidence, distribution, and possible control of diseases and other factors relating to health. So the founding father of our science is named John Snow. He was a physician in London, and he was studying cholera outbreak in London. And this was before germ theory. They didn't understand that things could be carried by bacteria. But what he did is he went from house to house, and he asked if they had any cases of cholera in the house. And if they did, he asked them where they got their water. And if they didn't, he asked them where they got their water. And he realized that by making a map, that almost all the cholera cases were getting their water from a single public pump. So this was before water was piped into homes. It was called the Broad Street Pump. And he thought that there was probably something in the water causing the cholera. So he went and he took the handle off the pump so it wouldn't work anymore. Everybody had to walk further to get their water, but the cholera epidemic abated, and he concluded that it was water that carried something that caused cholera. We know now it was because sewage was getting into the pump, and that was the problem. So this is the idea of mapping diseases and using it to make inferences about what the underlying causes are, and then what you could probably do about it to try to prevent the disease. Our other founding hero is a guy named Dr. William Osler. He was a Canadian physician scientist, and he said medicine will become a science when doctors learn to count. And that's making fun of physicians, which I think can be fun sometimes. But it's also, he's making the point that when a physician is doing their work, they're administering medicine, they're treating patients, but they're not actually doing science. They're using scientific knowledge that's been developed to help people today. But if they want to actually do something scientific to get new knowledge, they have to start working in a different way. And counting is one of the important things we have to do, is we have to count the diseases to be able to understand what we're facing. So the burden of cancer dramatically varies by organ site and geographic location. So what may be a common cancer in one country or in one part of one country may be a rare cancer somewhere else. Cancer doesn't occur at sort of an even rate all around the world. It varies dramatically, and that's because the vast majority of our cancers are caused by modifiable factors in the environment. So why would it be important to describe the burden of cancer? And I'm going to show you an example of describing the burden. This is a map at the county level that shows the incidence rate of lung cancer across the United States. So it's not a count of how many cases, it's a rate. It's how many cases per 100,000 people in that county. And if it's a small county, you can still calculate the rate per 100,000. And what you see is that in the red, that's areas where lung cancer is common, and the yellow is a place where lung cancer is uncommon. And you can see it's dramatically different. And red there is Kentucky. And Kentucky is where almost all of our tobacco is grown. And at this time, 40% of adults in Kentucky still smoke cigarettes. And so this is a huge burden of lung cancer in that community. And that's kind of what the whole country used to look like. In 1965, 60% of men were smoking cigarettes. If you look at, for example, Utah, the Latter-day Saints are opposed to smoking tobacco. Tobacco smoking rates are very low in Utah, so there's almost no lung cancer in Utah. And that is it's an important way of understanding the burden of disease. Because we know what causes lung cancer, I can easily explain why the map looks like this, but there's a lot of diseases where we draw the map and we have no idea why it looks like that. And so it's a place to start where we can get a hypothesis about what's going on. The other important factor is that if you are running a hospital and you're in Kentucky, you better be well stocked with thoracic oncologists who can treat lung cancer. Whereas if you're in Utah, well, there's not such a pressing need for people to study lung cancer. So these kind of maps are important for health services. And if you were a company, you might want to know this too, because if you're not at the state level, but at the country level, if you're designing therapies and you're focusing on lung cancer, you're going to have a lot of customers in the United States. You're also going to have a lot of competition because everybody else knows that too. If you're studying a rare cancer like small bowel cancer, which is really rare, and you have an agent that you think might be useful for small bowel cancer, you probably won't develop it because it won't be economical. You won't have enough patients. And so you might have to go and use a special program. There are special programs for orphan diseases that are too rare to make it economically viable to study their cure. And so you might be able to get support some other ways. So understanding the disease burden has a lot of ramifications from different aspects of our lives. This is the same map at the national level for lung cancer, and this map parallels what we know about tobacco smoking. US, Canada, Western Europe, China, Japan, all have very high rates relatively of tobacco smoking, at least at one point they did, because when you have a high rates of exposure to some carcinogens, that can last a lifetime. If you accumulate 20 pack years of smoking, which is smoking a pack a day for 20 years, your cancer risk is going to be elevated for decades, even after you quit. So same idea. We know that, for example, in Africa, tobacco smoking is quite rare. They do grow tobacco in a lot of places, but they export most of it, and so they're protected against lung cancer because they don't use the tobacco. And in India, tobacco is very common, but there's a lot more tobacco chewing in India than smoking. And so if we studied oral cancer, lip cancer, we would see that that's much more common in India. This is a figure showing how cancer rates changed over time in the United States. So in 1930, the state of Connecticut decided that they wanted to count all of the cancer cases that occurred in Connecticut, and they made it a state law. It's a reportable disease. When a physician diagnoses a case of cancer, they have to give some basics about the case to the State Department of Health. That's now the law in every state in the United States. Every state collects data on state cancer rates. And things have changed a lot since 1930. So if you look here in yellow, that's stomach cancer. In 1930, it was the most common cancer in the United States. It was a substantial cancer burden. But over the last, this only goes through 2005, but since 1930, there's been a huge reduction in stomach cancer. And this is something that I work on. We have a pretty good explanation for why this has happened. It has to do, one, with a bacteria that grows in most people's stomachs, at least at that time it did, called Helicobacter pylori. It wasn't discovered until 1985 that it existed. Even though it was in basically every human stomach, people thought the stomach was sterile, but they were wrong. And so that bacteria is slowly disappearing in the more modern societies. It disappears, and so we don't see stomach cancer in these areas. On the other hand, lung cancer was a rare cancer in 1930. It was almost never seen, and that's because machine-rolled cigarettes had only been recently become available. And even though people smoked tobacco, they smoked far less than they could. Once the cigarettes could be pumped out in a factory and were cheap, people smoked a ton more than they had before. And also, during World War I and World War II, tobacco was given to the troops. A lot of men became addicted to nicotine and became heavy, regular smokers. So lung cancer skyrocketed. It went up over 50-fold. It became 50 times more common than it had been. And in 1964, the Surgeon General concluded that tobacco caused lung cancer and made a report, and that changed the smoking trends in the United States. 1965-66 was the peak of smoking. It started to come down in men. It took longer. Women kept going up for much longer. But eventually, you could see that the cancer rates turned over, and that's because we had like 20 years of the smoking rates going down, so eventually the lung cancer rates went down. Okay, so again, these patterns really can help us understand and plan for how we can take care of cancer patients, and it can also tell us something. We do see trends like this that we don't understand sometimes, and when we see a change in a cancer, we have to start to think about what the causes are and whether we can figure out how to prevent this cancer from occurring or train more people to treat it. Okay, so how do we learn what causes cancer? But first I want to talk about what the word cause means. Okay, in this context, what we mean by cause is that there's a biological mechanism that links the exposure to the disease. People have lots of ideas about what might cause cancer, but if you can't explain the mechanism, you can't be very certain that you've really identified a cause. So with tobacco, we know that there's a lot of DNA damaging agents in tobacco smoke, and so that's part of the mechanism. With drinking alcohol is converted into acetaldehyde, which is a known carcinogen. It's also something that we call generally recognized as safe because the dose makes the poison. If you're exposed to a small amount of acetaldehyde, like when you eat yogurt, it's not an issue. But high doses of acetaldehyde in the local environment in your mouth because you're drinking alcohol can lead to cancer. So we need to have a mechanism to say something's a cause. And there has to be a predictable dose-response relationship. If you have a group of people who are only lightly exposed to a carcinogen, you should expect very few cases of cancer, even if it's a known cause. If they're heavily exposed, you should expect many more cases of cancer. And that's one of the hallmarks of a cause, is there's a dose-response curve that you can draw. What it doesn't mean is that everyone exposed to the carcinogen will get cancer. If you take one puff off a cigarette, you will not wake up with lung cancer tomorrow. That isn't the way causes work in disease. It's a probabilistic statement. We're saying that under certain conditions, under a certain exposure regimen, this is likely to cause cancer in some fraction of the people exposed, not 100% of the time. When I can tell you that it's common when you talk about causing cancer is you'll hear somebody say, my grandmother smoked for 40 years and she didn't have lung cancer. So I don't think tobacco causes lung cancer. And the answer to that is, well, no, your grandmother was lucky. And even heavy smokers, people who smoke 2 packs of cigarettes a day, only 10% of them will get lung cancer in their lifetime. One of the reasons is there's competing mortality. Cigarettes cause more heart attacks than they do cancers. And also, even some heavy smokers dodge the adverse effects, but that doesn't mean it's not the cause, okay? And another thought experiment that you could do is if I say, last year, 43,000 Americans died in automobile accidents, I don't think anybody's going to say, well, my grandmother was in a car accident, she was fine. You think about the logic. It doesn't mean that just because you have the same exposure, you're always going to have the same outcome. So we have to imagine that there's different scales of what we call sort of like potency, right? Okay, so when we're thinking about causes, we have to be careful. We're not expecting anything to be a one-to-one relationship. The only thing that's really a one-to-one relationship is some types of genetically induced cancer, where if you have a particular variant of a gene, you will get the cancer as long as you don't die in infancy. But that's about the only cancers that have that level of intensity of the association. Okay, so causes. So one idea is that you could ask people, why did you get cancer? So here's a doctor speaking with a patient. You can ask them about why they think they got cancer. And that's a question that nobody can really answer. And actually, we can almost never answer it in an individual cancer case, why did that person get cancer? Even people who smoke 2 packs of cigarettes a day can get lung cancer because there was radon in their basement and they spent their whole life breathing radon. So we can't always say why an individual got cancer. We have to talk about it as groups. People have a lot of ideas about why they may have been afflicted by cancer. They may feel bad about some behavior that they did and attribute it to that. They may feel angry at somebody who they think exposed them to something and they may attribute it to that. But talking with an individual cancer patient on their own is not a great way to understand what causes cancer because it's just hard for anybody to know. So what else could we do? Well, we can do laboratory experiments. I mentioned that basic science is an important part of the cancer research enterprise. And so if you have a hypothesis about a particular chemical, you could do studies in vitro with cells in a tube and see if it damages DNA or if it causes the cells to grow uncontrollably and so forth. And that's an important part of the process. It's not enough by itself. Another thing that we often do is we do animal studies. So many rats and mice have been sacrificed to our understanding of what causes cancer. You can expose rats to graded doses of a carcinogen, and these can be genetically identical rats that live in the same housing with the identical food, and the only thing that differs is the dose of the carcinogen. And if you do that, you can get nice dose-response curves, as I said, and that helps explain it. But if something is safe in a rat, should we conclude that it's safe in people? I would say no. People aren't rats. We have very different physiologies. And so things that happen in rats, they aren't perfect models for what happens in humans, but again, it adds to our understanding. So really to get there, we need to do epidemiology as well. And so epidemiology is studying free-living people, how they live their lives, and you use them to help you understand what causes cancer. So you get a lot of people are willing to volunteer for cancer research studies. We have a lot of generous people are happy to participate. So what would you do? If you got these people, can you divide them in half, have half of them not smoke and have half of them smoke cigarettes for 40 years so that you can prove in an experiment that that's what causes cancer? Well, of course not. It would be unethical to intentionally expose people to carcinogens in order to do this experiment. And many people believe that if you don't do an experiment, you're not doing science. And if you don't do an experiment, how can you really be sure about the results? And I agree that it's not ideal, but we can't. We can't do that. You can expose people intentionally to stuff that you think prevents cancer. I do nutritional studies where we've randomized people to get nutritional supplements to see if that prevents cancer. And that's okay, but you can't do that if you want to understand what causes cancer. So we have alternate strategies, and that's what we call analytical epidemiology, and it's an observational science. It's not an interventional science. So what we do here, in one example, is that you could sit at a hospital, and I have studies where I do this, and every single person who gets diagnosed with a type of cancer, you invite them to participate in your study, and those are your cases. And you ask them a whole series of questions about their life, and you take blood and urine and saliva, sometimes feces, maybe tissue biopsies, and you can do a lot of work to understand their exposure history by doing that. Then you get a group of healthy people, and you do the same thing to them. And then you compare the rates of exposure. So if this was a lung cancer study and you ask them about their history of smoking cigarettes, you'll find that the lung cancer patients have smoked way more than the healthy control group, and you can conclude that there's an association between the exposure and the disease. An association does not mean that it's causal. It means that you've detected a pattern that you think is important. Another way to do this, which has fewer problems, is a cohort study. A cohort study is you just collect healthy people, you ask them, you do the same thing to ask them all about their lives, and then you add time, and you wait to see what happens to people. So we'll do this, and for a cancer study, we'll usually collect in the neighborhood of 30 to 50,000 people and ask them about their lives, collect blood samples and different things for them, and then we'll wait to see who gets cancer and who doesn't. Now, it sounds a little harsh to do that, but that's just life. People go about their lives and things happen to them, and we use that to understand why that happened to them. But this is a very slow process, and you need huge groups of people. Lately, we're doing even bigger studies, hundreds of thousands of people, and we often follow them for decades. You're not going to do that to answer one question. If you have a cohort that big, you're going to answer many questions. I run a cohort out of my group where we've published over 500 papers from a single cohort because we've been able to address dozens of different cancer sites and multiple dozens of different exposures. I'll give you an example. So I've been using cigarette smoking as an example tonight. We already know that cigarettes cause lots of different kinds of cancers, but we had a huge cohort, and so we had an opportunity to make better estimates of that association. And it's a cohort that's called the NIH AARP cohort. It was run by NIH, and the AARP partnered with us. And this was a cohort that was created by my old boss, Arthur Shatskin. He conceived of the idea of making a mega cohort, and he said he wanted to recruit more than 500,000 people. This is incredibly ambitious back in the 1990s. And we partnered with the AARP organization because it's an affinity group, and when they put their name on it, people were more likely to listen to our pleas to participate in the study. It was a relatively low-impact study. They just needed to fill out a questionnaire every couple of years, and we got more than 500,000 people to participate. And we've been following these people ever since using those state cancer registries. They gave us their social security number, and with that we can find out if they've had a cancer diagnosis. And then we use a very interesting statistical method. A lot of my work is basically statistics and programming, but I had heard that for every equation you put in a talk, half the audience checks out. So I haven't put any equations in anything tonight. I'm just going to skip over the statistics, even though it's the main thing that we do. I don't want to bore you with that. Okay, and I also want to note that Arthur, who conceived of this study, died of glioblastoma before he got to see it come to a fruition and all the power that it could do. Okay, and so here's an example. This is 20 different cancers. I'm going to zoom in so you can read it. So lung squamous cell carcinoma is a subtype of lung cancer, and it's known to be the most affected cancer by tobacco smoking. And in this huge cohort of people, if you look at the top line, we had 285 women with this cancer that were current smokers. We had 7 who got the cancer who were never smokers. And in this cohort, about 40% of the women had smoked at some time in their life. About 20% were current smokers when they enrolled in the study. And by doing some math, we calculate what's called the relative risk. And we show that for lung squamous cell carcinoma, being a current smoker makes you 100 times more likely to get this type of cancer. It's a massive effect. And we then estimate what's called the attributable fraction, and that was 97%. That means 97% of all those cancer cases were caused by the cigarettes. We only can say caused because of all the other work that's been done. If this was the first time we had ever studied this exposure, we would never say it was caused by it. We would just say it was we were building evidence for the hypothesis. If you go to the other end of the scale, down here is colon cancer, and you can see, first of all, there's a lot more colon cancer. We had 321 women who were smokers who got colon cancer, 722 who were never smokers who got colon cancer, and here the relative risk is 1.3. So if you take the rate in the never smokers, multiply it by 1.3, it's just a tiny bit more colon cancer. Now, you may not have thought that colon cancer is linked to cigarettes. You have to have a huge study to be able to see that because it's a small effect. For colon cancer, we estimated that about 10% of colon cancers are caused by cigarette smoking. So that's how we can estimate quantitatively the effect of an exposure. But tobacco is a special case because it's so potent. There's very few other carcinogens that are that potent. One of the other ones that like that is human papillomavirus. It's 99.9% of all cervical cancers are caused by human papillomavirus. And in the future, we'll have next to zero cervical cancer because once we get everybody vaccinated against HPV when they're young, there will be no more cervical cancer. It will disappear. Because if you don't have HPV, you can't get a cervical cancer, and with HPV vaccine, you don't get HPV. So it's going to be a perfect prevention, but we have to wait about 100 years to process that through the population once we get to 100% vaccination rates. HPV vaccine also prevents cancer at least 3 other sites in the body, and those sites will also be protected against cancer by having HPV vaccination. Okay, so I've mentioned that we have lab studies, we have animal studies, we have these epidemiologic studies. There's a lot of argument about whether the epidemiologic studies, how strong they are, but who gets to draw a conclusion? Who gets to say this causes cancer? Well, the first person who gets to do that is you. All good data is public data. If you can read the literature, it's out there. Anything that's published using NIH money has to be freely available 1 year after publication, and we have a thing called PubMed Central where every paper that's published using NIH money, which is about 70% of the papers in the United States, are made freely available. Anybody can read them and draw your own conclusions if you feel like you're able to read that literature. So that's important, but there's also independent groups. One is called the International Agency for Research on Cancer, which is part of the World Health Organization, which is part of the UN, and they have a research group that does what are called monographs. They bring together experts from all over the world to review all of the public literature, and then they write reports about whether or not they think there's a cause of cancer. And they rank the level of evidence as causal, probably causal, possibly causal, definitely not an association, and unknown. Okay, there's another group that's run by the US government called the National Toxicology Program, Report on Carcinogens. They do the similar thing. When these groups draw a conclusion, what necessarily happens is nothing. If they make this conclusion, sometimes all that happens is there's publicity about it and there is no change in society. People may be made aware of the problem and maybe they change their behaviors, but there isn't necessarily anything that happens legally because of these conclusions. And so they're important, but putting them into action is a regulatory body's action. So I work at a research agency. We do not make regulations. If we learn something important about cancer and there needs to be something that happens because of that, it would be the Food and Drug Administration, the Environmental Protection Agency, or some other regulatory body in the United States that might take regulatory action. That's not for us to do. Okay. Is epidemiology the weakest link because we can't do randomized experiments? Yes, that's absolutely true. And that's because correlation doesn't equal causation. We're doing complex study designs. We're doing all sorts of ways of controlling it. We're doing very fiddly things to understand these associations. But in the end, we have to be humble and say, well, it's an association. So what we usually do in epidemiology is we do it over and over. And we do it in different contexts with different people, and we look for a consistency of the finding. Now, there's a lot of funny correlations out there. One is that ice cream sales and shark attacks are highly correlated, and it's not because sharks like a cream-filled victim. It's because in August it's hot and people are outside, they have ice cream, and they also go swimming, and so you get this funny correlation. And it's absolutely true that shark attacks and ice cream are not causally associated. And so we don't want to be a person who makes a claim about a cancer-causing agent for something like this, so we tend to be very careful about the conclusions we draw. Now, I've said that this is the best we can do, is to take all these different things and put them together, but there are people who espouse what's called evidence-based medicine, and people who like evidence-based medicine may say that without a trial, we shouldn't act, because you can't really be certain. So the clinical trial is the top level of evidence, and so they'll be very skeptical of anything that's done with the kind of epidemiology I mentioned that's observational. And this is something that people fight about a lot and professionally fight about it. And so people have made a lot of humorous papers to try to expose the problems with the idea that we have to have clinical trials. The British Medical Journal, every year around Christmas, they publish an issue full of humorous scientific papers, and they published one about parachute use and clinical trials of parachute use. And what they did was these two people, Smith and Pell, they searched all the literature to look for clinical trials on the topic of parachute use to prevent death and major trauma related to gravitational challenge, which means falling. And so they searched the entire world's literature, and what did they find? Well, there's never been a randomized trial of parachutes. And from an evidence-based medicine point of view, they said there's no trials. Therefore, the basis of parachute use is purely observational, and its apparent efficacy could potentially be explained by a healthy cohort effect. And so this was a joke, but it really makes the point that if somebody's telling you, well, there's no clinical trial, so I don't want to act, I don't want to do anything to change anything, you ask them. And their point was that individuals who insist that all interventions need to be validated by a trial should come down to earth with a bump. And they invited all those people to participate in the first ever clinical trial of parachute use. And we would do what's called a crossover trial. And that means first you get a parachute and half doesn't, and then you switch. Everybody, you switch exposures, okay? So it's funny, but it makes an important point that sometimes things are strong enough that we can make conclusions without trials. In fact, a few years later, a group decided, forget it, there's no parachute use trial, we're going to do a parachute use trial. And they carried one out. They got 25 people to volunteer for this parachute trial, and they published it just like a real clinical trial. They said, what's known? They said, basically, it's just observational data, and people jump out of planes with parachutes because they think it's a good idea, even though it's never been proven. There's at least a dozen people who've fallen over 10,000 feet without a parachute and survived. So we know that you don't have to have a parachute. You can survive without one. And so they went ahead, did the trial, and what they found is that there was no difference. They did half the people jumped out of the plane with a parachute, the other half jumped out with an empty backpack, and yet everybody survived. And there was a little problem in their trial, is that they didn't take off. So they just jumped off the wing of the plane. They definitely jumped out of the airplane, but it turned out that they had a problem, and as they said, they thought the problem is they didn't have enough people at high risk of disease. And this is lampooning the ideal that you have to have a trial, because even when we do trials, when we can, there's always somebody who says, well, you did the trial wrong. I would have used a different compound, I would have done it for a different amount of time, I would have used a different group of people, I would have dosed it 3 times a day instead of once a day. It's like you can never stop complaining, which is true, but for most people, we think, even without trials, even with observational data, non-experimental data, we can make decisions and we can take actions, and those actions have led to a huge amount of reduction in suffering. When the Surgeon General said cigarettes cause cancer, a lot of people stopped smoking, and a lot of lung cancer cases have been avoided because of that. So I think it's an important aspect of what we do, and we have to strive for the best evidence we can get, but we can't always get the best evidence we want. Okay, so I'm going to tell you now about this was my title. I've talked to you a lot about general stuff. I'm going to, just in the last few minutes here, I'm going to tell you a story about work that I'm doing to try to understand something that nobody knows if it's true or not. I still don't know if it's true, but here's what I did. And when I was in high school, I watched the TV show on KTCA called Nova, and I'm going to show you the beginning of the show. Let's see.
is a street opera in Linshan.
Okay, notice that she's got pretty good looking teeth there. So I was 13 years old and I saw this show and I couldn't believe it because it was these Chinese doctors and they were in a town called Linshan, and in that town at least 10% of all people died of esophageal cancer. There's no other place in the world that has a concentration of cancer like this. Esophageal cancer is a really nasty way to die, and it really stuck with me. But I went to school and I didn't study cancer, I studied other stuff. But I go to the NCI and I'm looking for a mentor to help me do a research project, and I meet this guy and he says, oh, I study esophageal cancer in China. And I was like, really? I said, I saw a show when I was a kid called The Cancer Detectives of Linshan. He said, yeah, that's where I work. And I couldn't believe it. And since after the show was made, Americans got involved in the research there. And here's this guy who's been working in Linshan for 2 decades. And came back to me, and for other reasons, I really wanted to work with this guy. And so I started working with him, and this TV show I saw when I was a kid came back, and I actually joined into this project that had started long before I was ever around. And the reason Americans are working in cancer in China and not just in the United States is something called public health diplomacy. And because of this guy, that's President Nixon, and he's eating with Zhou Enlai. And you can see that he's really good with chopsticks. He doesn't have the faintest idea what he's doing. And the look on his face like he thinks he's going to have to eat a bug or something like that. And Nixon had all sorts of problems, but he really hated cancer. He started what was called the War on Cancer, which elevated cancer funding dramatically. And he opened China up to different kinds of collaboration that we'd never had before. So first thing they do is they send athletes when you have this kind of public diplomacy, and our ping pong teams went to China and got their butts kicked because the Chinese were much better at ping pong. But we had cancer researchers, we had some advantages there, so they invited a bunch of cancer researchers to go to China, and China had beautiful maps of the patterns of cancer in China, and one of them was the patterns for esophageal cancer, and these scientists decided that that would be an important place to have a partnership. So they started working together in 1982, and in 1985 they started a huge study there. And when I got to the Cancer Institute in 1999, it was still ongoing, and so I've joined that study, and now everybody who started it has retired, and I've inherited this study, and I'm carrying it forward. And one of the things they looked at is they had this idea that having bad teeth might be bad for your cancer risk. And this guy was in a trial, and the main thing they did there was a trial. And my idea is that I could turn around and use the same data for observational studies. The trial had answered the trial question, and I wanted to reuse that data for other purposes. This guy has no teeth, and we're not worried, but this guy only has 2 teeth. And we wonder, what does that mean? Does having bad teeth cause you to have other bad health outcomes? And the first paper I ever published in Epidemiology was in 2001, and it was called A Prospective Study of Tooth Loss in Incident Esophageal and Gastric Cancers. And what happened is that all 30,000 people in the trial filled out a questionnaire that was read to them, and one of the questions was, have you lost any permanent teeth? And if they said yes, they asked them when they lost it, and then they had them open their mouth, and the interviewer counted the teeth they had in their mouth. And this data had been sitting around for 15 years. Nobody had ever touched it. And I came into the study and I said, well, that's kind of interesting. Maybe I could look at that data. And when I looked at it, I did these statistics that I won't bore you with, and I concluded that there was a modest association, that having lost a lot more teeth than your peers made it slightly more likely that you would get cancer in your esophagus. And as I said, this is a relatively weak association. It had barely ever been reported before. Somebody had thought of the hypothesis because they bothered to count the teeth. And I thought this was interesting, and then I went on studying other things, but it was really interesting to me, and I kept doing tooth studies when I could. A few years later, I published a similar study, but here I looked at death from all causes, death from upper gastrointestinal cancer, heart disease, and stroke. Again, I found evidence that people who have bad teeth are more likely to die from these certain causes, and more likely to die younger than people with good teeth. And then somebody said to me, well, that's just tooth counts. If you really want to do something, why don't you get a real dentist to do the exams? So I got a public health dentist from the United States to come with me, and we did a study on 800 people where we also did endoscopy on them to look at the state of their esophagus, whether it was healthy or had preneoplastic lesions in their esophagus. And he did a very intensive exam on all these people. And so we got much better dental health data than we had before. Same thing. We found pretty good evidence that having bad teeth was linked to having more preneoplastic lesions in your esophagus. So we're building the evidence that there's an association there. I went on to do this. I've published about 40 papers about teeth and cancer, and there's this very consistent pattern forming. Most recently, I published this paper with some colleagues in the United States where we were able to do, for the first time, untreated caries. We never had that kind of data before. Here we were looking at total mortality, and now we're going to go further and look at individual cancer types. And you can see this was published this year. So now I've got 20 years where I've been pursuing the same hypothesis using observational studies. Been doing lots of other stuff at the same time. But after all that time, I'm really convinced that at an observational level, I have pretty good reason to believe that having untreated caries and periodontal disease is going to increase your risk of getting cancer in your stomach and your esophagus. And that may suggest that the right thing to do is to make sure that people get good oral health care. It won't just help them with their mouths, it'll help them with their preventing cancer. But we don't have that mechanism. I said that we have lots of different aspects that we want to do to have a powerful conclusion, so I need to address how this mechanism could happen. And so in parallel, I've been doing that. I've decided that I think that there's something there, but I wouldn't swear to it. Other people might say my evidence is, so-so, you're getting somewhere. But what else can we do? How do we get to the mechanism? And there's something called confounding, and that's when you make a mistake. Confounding is when you think there's an association, but it's really caused by another correlation. If you go and study car accidents, you'll find that people in car accidents are much more likely to smoke cigarettes than non-car accident people who haven't been in car accidents. And you might come up with a hypothesis. It's that because they're fooling around with a cigarette or they're trying to light it and they get distracted or something, but that's not it. It's because people who smoke are much more likely to drink alcohol to excess, and it's really the fact that they're drunk is the problem for the car accidents. It's not the tobacco, and that's called confounding, and it's when you make a mistake in your causal pathway. So people said to me, well, tobacco and alcohol cause cancer, and tobacco and alcohol make people's teeth fall out, which is true. And so they said, it's just confounding. So what I did is I did all my studies, including people who had never smoked and people who had never drunk alcohol, so it couldn't be confounded. People said, well, when you have a bad diet, your teeth are more likely to fall out, and the bad diet makes you at higher risk for cancer. That's true, but it's too complicated to go into today, but I've basically shown that that can't explain what I'm seeing. Other people said, well, if you don't have good teeth like this guy, then you swallow partially chewed food, and that causes it to damage your esophagus physically. That's not correct. In fact, what happens is when you don't have teeth, you eat soft food. And so that's really not the problem. But then, what is it? Well, maybe it's the bacteria. The main reason that people have teeth fall out is because they get periodontal disease, and you get these bacteria that grow down in the crevices of your gum, and they eventually destroys the bone, and so the bone is gone and your teeth fall out because there's no bone to hold the teeth in. And that's what happens with untreated periodontal disease. And those same bacteria produce all sorts of weird metabolites, and some of those metabolites might be carcinogenic. It's also an intense inflammatory process. That's the reason your gums get swollen and red, is because you're having inflammation at the site, and that can have indirect effects. So the idea is I just need to measure the bacteria in people's mouths rather than count their teeth. And that's a great idea. But when I started doing this, doing the bacterial measurements was way too laborious. 20 years ago, I would have been in the lab for weeks to measure one person's bacterial communities. It's impossible to do it at the scale I need to do for an epidemiologic study. But the DNA sequencing revolution and what's called the Human Microbiome Project has fixed that. There's a huge revolution in the way we sequence DNA, and it's made it possible to take a sample from somebody's mouth, and with almost no effort, I can sequence all the bacterial DNA and figure out which species of bacteria are in a sample. It's relatively easy to do and inexpensive, and so now I can do those studies. I just have to have the raw material. And so I set out to do this, and I was trying to find a lab, and I was asking the lab about their quality control procedures, and most of them said, well, if we get sequence, then it worked. And I said, well, no, I think if you got sequence, you got sequence, but can you consistently get the same sequences? They said, no, we don't know. We haven't really looked into that. So me and some of my colleagues, we built a set of 96 samples, and we sent it to 15 labs, and we said, please tell us what's in this sample. What bacteria are in this sample? And what came back was a mess. About half the labs gave us the right answers, and half the labs didn't get it right, and it was because the science was so new, it was being done inconsistently. So we did what's called the Human Microbiome Quality Control Program, and we developed systems to make people so that they could accurately sequence DNA samples for bacteria. Now this is far afield. I wanted to study cancer, and I wanted to study how the mouth leads to cancer, but the technology wasn't there for me. When the technology was almost there, I found it couldn't be applied correctly, so I got involved in figuring out how to do consistent sequencing. Now I can do what I need to do. Okay, but what sample? I'm talking about teeth falling out. There's many different surfaces in your mouth, and they have different bacteria, slightly different bacteria grow in your palate versus your teeth versus in the subgingival crevices, which don't have much oxygen, so you have anaerobic bacteria down there. And so there was all these different things, and you know, I said I like to collect 20 or 30,000 people. Can I really go in and sample all these different samples in somebody's mouth and wait for 20 years? No, I can't do that. But luckily, Scope can do it for me. So in the 1990s, people realized that if you swish mouthwash in your mouth and spit it into a tube, you get plenty of human cells, and you can sequence the human genome out of those cells. So some of the epidemiologists I work for were going around having everybody swish Scope and spit it back into a tube, and they put it in the freezer. We had 100,000 of these samples in our freezer, and I was able to show that the Scope mouthwash sample gives a nice average of all the bacteria that are in all these different habitats in your mouth, and that works. So now I went back, I have 100,000 people who spit into tubes in 1995, and I know who got cancer and who didn't. They're all Americans, and so what do we get in America? We get lung cancer at high rates. So I did a lung cancer study, and here it is, a study that was just published last month. It's the first big study that my student, former student, Emily Vogtman, led in a huge group of people that we needed with expertise in DNA sequencing, statistics, and all sorts of different things. And we did the first study where we can show that the bacteria that are in people's mouths can predict who's going to get lung cancer 10 or 15 years in the future. Now, it's not such a good prediction that I could take somebody here and look at your mouth and say, well, sorry, I hate to tell you, you need to get in a lung cancer screening program. It's not that kind of prediction, but I could tell a pretty good group. I could separate the group that's at more risk from a group that's at less risk, and we're working on it. That's the first time we've done this, but the goal is to do it better. Now we need to get down to we used a really high level of taxonomy for the bacteria. Now we need to get down to the sort of the subspecies level for the bacteria. And we think if we do that, we can come up with a way of separating out these bacteria, and that'll be helpful in understanding this connection. And this is not something that's driven by tobacco. It happens to be that most of lung cancer cases smoke tobacco, but maybe this separates out the people who smoke tobacco and don't get lung cancer from those who smoke tobacco and do get lung cancer. And then the next question will be well, what do those bacteria actually do? Once we figure out the identity of them, then I need a microbiologist to help me figure out what their life is and what they're doing. So that's an example of the kind of work that I do. The Connect for Cancer Prevention study is one of these cohort studies that we're starting now. It's something that I'm helping to create. We're going to collect 200,000 Americans. We're going to have access to all their medical records because we're doing this through HMOs, and Health Partners in Minneapolis is one of our partners. And so members of Health Partners are being asked if they'll volunteer for our study. Also for the Sanford Health System in South Dakota is another one of our partners. And they're being invited to join. And if they join, we give them an app on their phone. And about every 6 months, we send them a questionnaire on their phone, and they just fill it out on their phone. And then we ask them to come into the clinic about every 3 years, and we collect their blood and their urine. And I send them a kit with Scope, and they spit it back into a tube, and they mail it back to me so I can study their mouths. And my colleague Emily is sending them a fecal occult blood testing card so we can have a sample of their feces in the freezer, and we'll be able to study the bacteria in their feces later on. And so this is a big ongoing study. It'll be about 10 years before we can really do anything with the data, which is when I'm hoping to retire. So my goal is to get this in place, and then my students can can take it over and somebody else can actually use this, because I benefited from stuff that people did when I was in high school, and so now I need to set it up for the next long-term studies for people who are going to come after me. These are the participating centers that we have right now. Okay, I think I explained to you what we were going to do. Okay, so my center, as I said, we're a federal government research agency. My group just does epidemiology. There's 300 of us in our division. There's 50 in my branch. And we do all sorts of different studies, and this is one of the things that your federal tax dollars goes to. So thank you, everybody who's paying their taxes. And if you like this, then you can think that maybe your taxes are coming to me. Thank you very much. And that's the end of my story. Thank you.
Thank you, Dr. Abnet. We do have time for questions. We have several options. If you raise your hand, I will bring the microphone to you. We are recording this so that it is available to other people, so we do need you to speak into the microphone, or we do have microphones in the front. So questions?
Do you think that vaping will cause a new wave in lung cancer?
No, not directly. Nicotine does not appear to be the causative agent for cancer in tobacco. It's the byproducts of burning the leaves and not the nicotine itself. So I don't think it will have a direct effect. We know that many people have switched to nicotine replacement therapies to try to quit smoking. A good friend of mine grew up in India chewing tobacco for years. He can't give up the nicotine, so he sucks lozenges. I think direct exposure to nicotine might have some disease effects on the heart. I think there's other stuff in some of the vape liquids because they weren't well controlled. A lot of vape liquids used to have artificial butter flavoring in them, the same thing they put in microwave popcorn that causes lung damage. But I don't think it's going to cause cancer in the lungs directly. It's regulated by the FDA as a public health issue because what we know now is that the vast majority of teenagers, their first exposure to nicotine is in a vape. And almost all of them that end up smoking cigarettes go through vape. They get addicted to the nicotine through the vape, and then many of them will switch to smoking tobacco, and then they'll get the risk that way. So it's a big issue. In the US, we treat it as a public health hazard for now, but in some other countries, they promote it as a safer alternative to cigarettes. It's a safer alternative. It's probably not a safe alternative, but I don't think it's going to be lung cancer. If somebody just vapes, I don't think their risk of lung cancer is different than non-vapers. Assuming they vape nicotine.
So once the potential bacteria is identified, would probably the next step being, well, why does this bacteria live here? Is there something affecting some people that allows them to live for it?
Yeah, absolutely. So I mentioned the story of Helicobacter pylori, which is this bacteria in the stomach, and it was discovered in 1985, and nobody knew that there were resident bacteria in the stomach at that time. The guys who figured that out got the Nobel Prize for figuring out that this bacteria lived there. But what they had found is that it causes ulcers. So peptic ulcers are caused by this bacteria. And something that people may not realize is that up until that time, peptic ulcers was in the top 10 causes of death around the world. People died from bleeding ulcers all the time. If you watch old movies, you'll see people who are drinking milk and eating mild diets and stuff, and it was so common for people to have this peptic ulcer disease. It took about 7 or 8 more years before people realized that it wasn't just causing ulcers, it was causing cancer. And it took forever. We still don't really know how that works. We don't have a good animal model. It took about 20 different species of rodents before they could find a species of rodents that you could get this bacteria to grow in them. It happens to be Mongolian gerbils. We don't really know why, but that took a long time to find animal models. But that's the idea is that if we find a single bacteria, maybe that we can understand that individual bacteria. But I think we also have realized that bacteria don't grow as individuals. They grow in biofilms. It's an ecology. And so it could be that it's because of 7 different bacteria and how they live together that we really have a problem. And that was too hard to too hard to study, but now with this new DNA technology, we may be able to study it better. Yeah.
First of all, thank you for coming here, Dr. Abnet. Really enjoyed your talk. I have a question more related to the bioinformatics. So with bioinformatics being an ever more increasing tool used in research, where data can be compiled and displayed and presented by researchers, what ways does the NIH do to champion these ethical practices?
So I can tell you in our group, so I said we're a staff of 300 in our division. And for bioinformatics, 10 years ago, we didn't have any bioinformaticians. We did have people who were interested, who self-trained because they were older and they wanted to learn how to do this. To do my microbiome research, I convinced my boss to let me hire a bioinformatician so I could have somebody who could process the data for me because it was not something any of us knew how to do. The other thing our division did is we've now created a section on data science. So we've hired a chief data scientist, a data officer, who's not a cancer researcher. They're not even really a health researcher, but they know how to use these databases and to use complex data in a way that we never could before. So we have a whole new field that we're bringing in to make this possible. Even more important, I think, than using the data is that they're helping us share the data. So in the past, our data sharing was really kludgy. You would send me an email and ask me if I could give you the data, and I'd say, sure. Who's your lawyer? And I would have to have your lawyer talk to our lawyers, and we'd have to do a data transfer agreement because there was, personally identifying information that somebody had, and so I couldn't just give it to anybody. And so if you weren't part of a big institution, it would take forever to do this. What we're aiming for is having a data platform where we can just give you a user code and you can come on. I'll knock off all the variables that I say you can have that are not sensitive, and then you can redo whatever I did and do it your own way. So within a few years, we'll have this data platform so that people anywhere can come on. We'll also provide you with all the cluster computing power you need. The government will provide that for you. And so we'll have real data sharing. And that's really important, especially in these new fields where we're just figuring out how to analyze these piles of data. And clearly people have better ideas on how to reanalyze it. We want to make it as frictionless as possible. But I can't just put it on a website. I have to have, the data controls are complex. And we're facing a countervailing problem with the general data protection regime in Europe, which is forcing people to be sharing less data and letting people try to control their own data more closely, which I understand the benefits of that. It's making our lives as researchers more complicated, but these data platforms may solve it for us. So that's what we're doing right now.
Just building on to that, where do you see our generation in terms of the war on cancer? What accomplishments will we be able to make?
Well, I think there's a lot of, there's tons of room for improvement. One of the things we have to recognize is young people today, probably 40% of you will have a cancer diagnosis in your lifetime. Partly that's because people live longer. So making it to, we have a lot of people who make it into their 80s or older. And so the longer you live, the more likely you are to have a cancer diagnosis. But for a lot of cancers, we're doing better, and people are surviving longer or getting cured. One of our goals is not always to cure. It can be to just hold it at bay. So you might still have the cancer, but it's not going to kill you. You're going to die on somebody else's service is our idea. You've got to die from something. And if the goal for cancer is to have as many of those people as possible die from a heart attack, right, or something else, because then we've held it back as long as we can. So there's a lot of room to improve. I think vaccination is going to become a bigger part of it. We've just now figured out how to prevent all cervical cancer. It also prevents penile cancer and anal cancer and oral pharynx cancer. Almost all cancer at the back of the throat now is caused by human papillomavirus, and this vaccine is going to fix all of those problems once we roll it out. We have ways to go because some parts of the world, they can't afford the vaccine, or we have the wrong HPV types. So I do work in Malawi. There's an HPV type that causes a lot of cervical cancer there. It's not in the vaccine. because that type doesn't affect wealthy countries. So we need better vaccines so we can more quickly adapt them to local contexts. That's an important thing. And I think that there's more work to be done on all the traditional ways of treating cancer. One thing I didn't talk about tonight is we do a lot of early detection research. We're developing new ways of finding esophageal cancer when it's still small, and we can do endoscopic surgery, remove just a little blip, and you go on with your life and you're fine. And that's something that we've figured out how to do at scale. We can only do it in places where the disease is common because you have to have a lot of cancer to make it worth bothering people. If you screen 10,000 people to find one tumor, it's not going to work. So we have to find ways to target it to the right people who are really at risk for that disease. And so we have new genetic prediction methods, and then we know about their lifestyle, and we can put it together, and we can do risk scoring, and then invite the people in for a personalized screening regime. So there's a ton of stuff for people to do. Our institute is huge. We have, I think, 5,000 people in the National Cancer Institute, and we're growing regularly.
Additional questions?
What studies are currently ongoing in regards to cancer?
I'm sorry, what?
What studies are currently ongoing in regards to cancer?
For which cancer?
What studies are currently ongoing in regards to just cancer in general?
Oh, okay, I'm sorry. There's literally thousands of individual studies going on. And there is at cancer.gov, there is a ton of information about different cancer types, and you can see the vast majority of cancer research is funded by the National Cancer Institute. There is a lot of money from industry, but that's very targeted towards therapeutic applications. But from a broader perspective, much of the research that's going on is funded by the National Cancer Institute. And there's each of the types of research that I've mentioned today there's just a huge number of different types of studies going on all the time at many different centers. The other thing that the National Cancer Institute does is we fund what are called comprehensive cancer centers, and these are sprinkled throughout the country. There's one in the Twin Cities, and the comprehensive cancer centers are a place that provide not only high-quality care, but they help develop the research infrastructure so that we can have more people doing cancer research. So that's a really hard question to answer. Just at my institute, we have 5,000 researchers doing cancer research, so it's a big undertaking.
Please join me in thanking Dr. Abnet for a wonderful presentation. A couple last items for us. Dr. Abnet, please accept, hopefully not violating any National Institutes of Health, a copy of Leonard Ford's Chemical Magic book that was revised by Wynne Grunmeyer, one of his former students. So please accept that. And because we are the Department of Chemistry, or Biochemistry, Chemistry, and Geology, your very own test tube pin to remember your time here at Minnesota State Mankato. Upright, yes. My son saw mine and it was not in the right position, so he did adjust that today. A couple last items. I want to thank Chris Kortz, the administrative assistant for the Department of Biochemistry, Chemistry, and Geology. This event would not happen without all the work she does behind the scene to help with that. So Chris Kortz, thank you for your work. Also, thanks to all my colleagues who encourage students to come, and especially Trent Vorlachek and Becca Mohn for their help and support of the Leonard A. Ford Lectureship. And thank you to our chemistry and biochemistry clubs who had the privilege and honor of talking with Dr. Abnet during lunch. They are hosting a reception outside the auditorium here, so please come and join a few things. And then when you get home, please do make sure you brush your teeth. And have a safe journey. Good evening.